Hi, welcome back for this session of review of lesson 3. The next question that we have here, asks how is the meaning of a theory in science different from the everyday use of the term? How is the meaning of theory in science different from everyday use of the term? So the meaning, or in science, theory is a proposed explanation for a wide range of observations and experimental results that are supported by a wide range of evidence, while in everyday conversation it means a speculation or something that is imagined or not to be true. So you can see that from this it is very clear that a scientific theory is in fact a proposed explanation of a wide range of observations and experimental results. So these should be observed or should be experimental results, which means it has to have gone through a scientific method that is supported by a wide range of evidence. Like, for example, we know that the theory of evolution is in fact has enough evidence. It has enough evidence to support it. It has enough evidence to support it. When we are talking of enough evidence, we are basically talking about scientific explanations which might have been acquired through observations of fossil record. It should be of fossil record. We have genetics. So study of genetics has also highlighted that we have these continued changes of changes in gene makeup of organisms. While in day-to-day conversation, it is just a speculation or something that is imagined. For example, someone may say, "Oh my God, what you are saying is just but a theory," which means to him, you cannot put it into practice. A theory should be something according to our normal day-to-day use, something which is believed to be untrue. So let's go to the next question here. The next question: how do a hypothesis and theories related? How do hypotheses and theories related? So a hypothesis, as we know it, leads to a testable. A hypothesis leads to a testable prediction of what would happen if hypotheses are valid. But theories are proposed explanations for a wide range of observations and experimental results. So that is supported by a wide range of evidence. So the results or the outcomes of a scientific experiment in testing hypotheses will lead to them being significant or non-significant which brings about theories. So theories can only be acquired if we have hypotheses. If we don't have hypotheses which would be tested, then we cannot come up with theories. If we don't have hypotheses, then we cannot come up with theories. The next question: how do light microscopes differ from electron microscopes? So we have many examples of how they differ here. So we are having here light microscope, we have here light microscope, and here we are having the electron microscope. The electron microscope. So one is techniques are simple here. Here techniques are simple. Here there is high level of technical skill needed in order for you to use it. Then another thing here is specimen preparation normally takes few minutes or few hours. Specimen preparation takes few minutes or few hours, while in electron microscope, specimen preparation may take several days. It might take several days. Another thing here is, here we will be observing live, we can observe live or dead organisms, but on the contrary, in the other case, or in case here of electron microscope, we must observe dead or dried specimen. So surface view of the specimen here is poor in light microscope, but good, and it has details when it comes to electron microscope. Light rays are used here to illuminate. So here a beam of light is used to illuminate the specimen, while in the other case here we are using electron beam. So a beam of electron. Then we have condenser, objective, and eyepiece lenses are made of glass here. Here they are made of electromagnets. Then light microscope allow resolution which is lower in comparison to the electron microscope. Light microscope, the image formation depends upon light absorption in different zones of the object, while here the electron microscope is due to electron scattering. Image is seen by eyes through ocular lenses. No screen is needed here, so we don't need any screen, but image here is received on a zinc fluorescence screen or a photographic plate. Then no vacuum is needed for light microscope. No vacuum is needed, while here you will need a vacuum. This one is very costly. This one is very costly. Very costly. This one is cheap. But those are just but a few of that we know, a few that we have here, but they are enough. The next question: why are computer models used in biological studies? Wow, what a question. Why are computer models used in biological studies? Imagine you want, for example, to explain about something like I want to explain about, for example, I want to talk about the entire biosphere, or I want to talk about energy flow. energy flow in the ecosystem. It's impossible for me to do this because these areas are wide. So the only thing that we do, we don't know the number of organisms that exist in an ecosystem, but we will take samples. Then from these samples we can build models like to come up with food chains. To come up with food webs. To come up with food webs, and so forth. So we say computer modeling is used because they can help scientists study complex systems on much larger scale. So that is one reason. Another reason why we use models is that they can also be used. They can be used. So we say they are also used when actual experiments are not safe or ethical or practical. So if the experiments that we want to carry out are not safe, maybe for our health, or it is risky in terms of ethics, then we would resort to models rather than go into those tricky parts. So how does molecular genetics add to our understanding of genes? How does molecular genetics add to our understanding of genes? So directly here, molecular genetics is the study and manipulation of DNA on a molecular level. So our understanding of the DNA molecule has led to many technologies like genetically modified foods, transgenic plants, animals, and even replacement of faulty genes. So entirely new areas of biology have arisen from combining molecular genetics with computer technology. Through computers, entire DNA sequences and genomes of humans and other organisms have been found. And after finding them, then these are saved in databases. They are saved in databases of computers, and they can be compared from one another. So we have databases for different genomes. Of organisms. So what happens here is, in case we want to find any difference or similarities of one gene to another of another organism, we will go to the database, and from the database we will be able to look at any similarities or differences. Or how one gene acts in one organism can determine how it will act in another. Next question. Viruses are smaller than cells. What type of microscopes could you use to study them? Explain your answer. So, electron microscope. Why? Because it has a higher magnification and resolution. It's very simple. If we would resort to one which wants to show us something which is very small, then we would resort for the one which will give higher magnification. Then provide an example of how technology has helped biologists gain a better understanding of life. So a good example is basically data mining. A good example here, so a good example is data mining, whereby searching computer databases, a biologist can find patterns, similarities, differences in biological data. With these details, a scientist can explore more of the diverse samples. A scientist can explore more of the diverse samples. So by exploring more of the diverse samples, then he will get a better understanding of life than if he didn't have this information. So we can say information is just at the click of a button. Information is just at the click of a button, and this is advantageous to the scientists. So in the next video we will look at chapter 5. We will be looking at the chapter 5 formative assessments.